Hello everyone. Today's topic is the things you should know for antigen presentation. So in this section, we know some things about for antigen presentation. Later, we will learn the detail for antigen presentation. So what is the function of MHC? So we know the history for the foundation of MHC, right? So at the beginning, the scientists found MHC because transplantation rejection. So they think this antigen decide their transplantation rejection. But later we know actually in our body, because not all the people need to transplantation, right? Actually, the real function for MHC is to present antigen. So that's class we know that the class 1 molecule or class 2 molecule, they present small peptide on their cell surface, and after that, the TCR recognize this peptide. So that means the real function for MHC is to present antigen. And after that, TCR can recognize this antigen. So after that, the TCL can be activated. Then we can start immune response. So for the antigen presentation, before we learn antigen presentation, we should know what is APC. APC, that means the antigen presenting cells. So here, so any cell, they have this class 2 molecules. So that class we know the distribution for MHC2 molecule is very limited, right? So they express for their special cells, such as macrophage, dendritic cell, and B cell. So this cell, they express class 2 molecule. So they have these cells, they have B7. Do you remember B7? So they gave their T cell the second signal. So these cells, we call it professional APC, professional antigen presenting cells. So now from the concept, any cell can present peptide. We call it APC, right? But actually no. Only those the cells express class 2 molecule and also B7. These kind of cells, we call it APC, antigen presenting cell. But other cells, now we know for class 1 molecule. Did you remember the distribution for class 1 molecule? They're all nuclear cells, right? That means in our body, almost all the cells, all nuclear cells express class 1 molecule. And this class 1 molecule also present antigen, right? So these cells, we don't call it APC, but they have the function to present antigen. So we call this kind of cells with class 1 molecule is target cells. For example, the virus infected cell or the cancer cell. So let us see one experiment. So this experiment shows us the antigen presentation or antigen processing. So from the upper one, so this is the macrophage. And this macrophage first fix. So after fix, this macrophage just remain their structure. So they are not alive cells. So if we give these cells a complete antigen, and after that, this macrophage cannot present this antigen. Negative, right? And no T cell activation. And the second experiment, so still this macrophage, and we don't fix it first, incubate with the complete antigen. And after that, these cells was fixed. Then they can make this T cell activation. So now in this experiment, we know. So something happened in the APC, right? In this macrophage, the antigen may be degraded, and after that, present. Then the last experiment. So same, the macrophage was fixed. Okay, still make this structure, and this cell is not alive, right? It's not a live cell. And after that, incubate with the small peptide, incubate with the small antigen. And this peptide binding this macrophage. After that, make this T cell activation. So this experiment shows us the antigen was processed and present on cell surface. Then the T cell can recognize this peptide, but the T cell cannot recognize the complete antigen. And also, this T cell must recognize the antigen. This antigen must be processed and present on cell surface. So this experiment shows us this antigen processing is a metabolic process. The antigen was digested into small peptide, then present on class 1 molecule or class 2 molecule. So what is antigen processing? Antigen processing means a protein antigen is degraded into peptides by a sequence of events. So later we will learn these events. What is the antigen was degraded and present on cell surface. And what is antigen presentation? The degraded peptide then associated with MHC molecule. So we know the T cell only recognizes the antigen presented by MHC molecule. So the T cell cannot recognize the free antigen. So this peptide binding with MHC molecule and present on cell surface. And after that, the T cell can recognize this MHC molecule present antigen. And this experiment shows us the T cell recognize the antigen as the self-MHC restriction. So did you remember why we have this self-MHC restriction? Because our T cell pass one strict examination, right? What is it? Positive selection, right? During the T cell development in the thymus, the T cell passed the first examination, we call it a positive selection. So from this experiment, we can see the mice infected with one kind of virus. And after that, the mouse can have their specific toxic T cell. We call it CTL, cytotoxic T lymphocytes. And in the right one, so no antigen. So this lymphocyte cannot. So this lymphocyte cannot recognize the antigen. So no virus-infected cell lysis. In the middle part, so we have the CTL, and we have specific antigen, and the virus-infected cell was killed by CTL. And in here, in the third experiment, now the MHC is different. MHC is different, so the CTL cannot recognize this peptide. So this we call it the self-MHC restriction from this experiment. So these two great scientists also got the Nobel Prize in 1996 because they found the antigen recognition by CTL exhibits MHC restriction. So for antigen presenting cells, we should, what is antigen presenting cell? As said, they must express class 2 molecule. So for dendritic cell, macrophage, and B cells, they have class 2 molecule. Also, they have B7, this molecule. So this kind of cells, we call it professional antigen presenting cells. And other cells, such as the vascular endothelial cells and the systemic epithelial cells, so these cells, they also can express class 2 molecule, but they don't have a special function present antigen. So these cells, they express class 2 molecule. We call it antigen presenting cells, but their function is not special to present antigen, and we call it non-professional antigen presenting cells. So if we want to know the antigen presenting, so now first we know what is endogenous antigen, later we know what is exogenous antigen. So for endogenous antigen, give you the example such as the tumor protein or virus or some bacteria produced inside the cells. So what is endogenous antigen? That means this antigen exists inside the cells. So inside the cells, they can produce new proteins. They can produce inside the cell. So now the cells think they were the endogenous antigen. So let's see, compare. If you not understand, it's okay, don't worry. So here, what is exogenous antigen? So originating outside the cells. For example, some bacteria exist, outercellular bacteria. For example, here, this is the outercellular bacteria. So these grow, the multiplication, proliferation grow outside. But after they engulf, you see internalized or phagocytosis by the macrophage. They come into, then this is just this antigen. They don't change, they don't produce new product, they don't grow inside the cell. So now they come into the cell and degrade into small peptide, then present on cell surface. So they already exist outside of the cell. Then they were engulfed or internalized by the cells inside the cell and degrade into small peptide, and we call it exogenous antigen. But endogenous antigen, someone exists outside, such as this virus and or some intracellular bacteria. They were foreign things, right? They exist outside, but they invade into the cells. Inside the cell, the virus can grow, right? Can survive become 1 to 2, 2 to 4, and they have their new virus production. So this kind of virus, we call it endogenous antigen. And also tumor protein. So tumor, actually, the cells is become malignant. So this antigen exists inside the cells. So I give you the examples. So some dead virus. So if engulfed by the cells, is it an endogenous antigen or exogenous antigen? So for the dead virus, because this dead virus, they exist outside of the cell, right? And engulfed by the APC. And they cannot grow inside because they already die, right? So they were the exogenous antigen. But if a living virus, they invade into the cell, inside the cell, and they grow and they become 1 to 2, 2 to 4, they produce new virus. So this kind of virus, this kind of antigen, we call it endogenous antigen. So difference between the endogenous antigen and exogenous antigen, because of whether they can grow inside or whether they can produce new protein inside the cell. So this is the difference. Because why I introduced the endogenous antigen and exogenous antigen? Because later we know they have different pathway to present by MHC molecule. Normally, the endogenous antigen will present on class 1 molecule, and exogenous antigen will present on class 2 molecule. So okay, so this section I just introduced some concepts. So you should know what is antigen presenting cells, the concept for antigen presenting cells, and what is the endogenous antigen and what is exogenous antigen. They were different. Because they are different, later we will learn the different pathway for antigen presentation.